Welcome to Learning Management System, Commissionerate of Collegiate Education, Vijayawada, Andhra Pradesh. Myself, Indravati, Assistant Professor in Biotechnology, KVR Government College for Women, Autonomous, Kurnool. Dear learners, the subject Recombinant DNA Technology is made easy for you. Do follow all my video lessons to cover different topics related to this subject. Dear learners, today we will be talking about screening methods, and in this session you will be learning about the significance of the screening methods and how these methods are used to select the transform cells, and you will be knowing about marker genes, reporter genes, and insertional inactivation and blue-white screening methods. Dear learners, let's study about the screening methods in detail. Screening methods are used for the selection of the transformed cells. You know that the transformation frequency is very low in genetic engineering experiments. Only a very few cells they get transformed. They are competent to take the transgene, and the remaining cells they are non-transformed. So you have to select the transformed cells from the pool of non-transformed population. So during this, the marker genes they play a very important role for easy detection of our transformed cells. So the marker genes they are introduced along with the transgene during transformation. They help in assessing the success rate of genetic transformation study, and they are used to determine whether our foreign gene has been successfully inserted in our host organism or not. So you can easily detect. They are of two types. They are selectable markers and scoreable markers. So if you see the gene construct, that is how the marker gene is inserted along with our foreign gene. So the selectable marker is also flanked with this promoter and terminator, and this one is clubbed with our desired gene. This purple color one is the desired gene. So this desired gene is flanked with this regulatory genes, and whenever you are using this, that is the desired gene is clubbed with the marker gene, whenever the target gene is expressed, the marker gene will also be expressed in the meantime, and in this way by this visualization of this marker gene product, you will be able to easily detect the transformed cells. So first coming to the first type, selectable marker genes. A selectable marker gene, it is a kind of DNA sequence which is inserted along with the foreign gene, and it helps in distinguishing the transformed cells from that of the non-transformed cells. Here two types of examples they are taken. One is the antibiotic resistance gene and another one is the herbicide resistant gene. The first one is the antibiotic resistant gene, neomycin phosphotransferase NPT2 gene. So this is the selectable marker which is isolated from E. coli bacteria, and this enzyme it encodes aminoglycoside antibiotics such as kanamycin, neomycin, and the normal plant cells they are sensitive to these antibiotics, whereas the transformed cells, the recombinant transformed cells, they are resistant towards these antibiotics. So expression of these NPT2 in the plant cells, it results in the synthesis of the enzyme which detoxifies this antibiotics and gives resistance property. So in this figure you are able to see that the transformed cells are having the resistance property and they are able to grow on this antibiotic containing media, whereas the non-transformed cells they are susceptible and they are dying. So it is very clear from this picture that the non-transformed cells are susceptible, whereas the transformed cells are having the capacity to resist and they are able to grow on this selective antibiotic selective media. Coming to the second example, herbicide resistant genes. So the example taken here is EPSP synthase gene. It is nothing but enol pyruvyl shikimate phosphate synthase gene. This gene is isolated from the mutant strains of Agrobacterium, and the transgenic plants which are having this marker gene, they are having the ability to resist this herbicide, in particular a broad spectrum herbicide called as glyphosate. So the transgenic plants containing this EPSPS gene, they can be selected on glyphosate containing medium. So here you are able to see in this picture the herbicide resistant transgenic coffee plant and the non-transformed coffee plant. So when you spray this herbicide, glyphosate herbicide, the resistant plants, they are green, whereas the susceptible plants, they are prone to this, easily prone to this herbicide. That is what is observed. Coming to the next one, that is the insertional inactivation is also a method which is used to screen the transformed and the non-transformed cell. In this approach, here the foreign gene, that is the recombinant cells, they will be appearing white in color, whereas the non-recombinant cells, they will be appearing blue in color. So what you are doing in this method is, in the LAC-JUT gene, the marker gene here used is LAC-JUT gene. This gene is having the ability to produce an enzyme called as beta-galactosidase. So this enzyme, in the presence of the substrate X-gal, it produces a blue color product. So whenever you are inserting a foreign gene in this marker gene, the marker gene, it gets inactivated. So this method of inactivation is called as insertional inactivation. So by inserting the foreign gene in the marker gene, it loses its property. So what property it is losing means the property of the production of this enzyme beta-galactosidase. So this enzyme is no more produced. So no enzyme means no reaction and no substrate reaction. It doesn't react with the substrate, and you will be getting colorless. No color product is formed. So in this manner, the transformed cells, that is the recombinant cells, because they are having the foreign gene inserted in this LAC-JUT gene, they produce white color colonies, whereas the non-transformed cells, because there is no gene is there, no foreign gene is there, it will be producing the functional enzyme beta-galactosidase. It will be reacting with the substrate, and when you plate it on a medium, you will be able to see blue color colonies. So that is what is represented in this picture. The media which is containing the X-gal substrate, all the white colored cells, they represent the recombinant cells, whereas the blue colored colonies, they represent the non-transformed cells. Coming to the second type of marker genes, reporter genes. The reporter genes, they helps to quantify the level of expression of the desired gene. That is, here you will be able to select the transformed cells, and also the additional property is you can be able to estimate how much that particular gene is expressed, the level of expression. You can quantify the expression of the desired gene. So that is the additional function of this reporter gene. So it encodes visually identifiable characters, can also be seen by means of these reporter genes. So here you will be studying two examples. One is GUS gene and another one is GFP gene. So this GUS gene, it encodes an enzyme called as beta-glucuronidase. This GUS assay, it is easily detectable method. By means of just by visualizing the cells, you can easily say that these are the transformed cells and these are the non-transformed cells. So they appear in blue in color. the transformed cells. The beta glucuronidase, it is isolated from this bacterium E. coli, so which can transform the colorless or non-fluorescent substrates into colored or fluorescent products. So by means of histochemical analysis, and when you use the substrate X-gluc, that is 5-bromo 4-chloro 3-indoyl glucuronide, or the transformed cells, because they contain that particular enzyme beta glucuronidase, they produce blue colored product, whereas the non-transformed cell, they show colorless, they will be appearing in colorless form. So this that is the level of expression of that particular transgene can be quantified by spectrophotometrically or by means of histochemical analysis. The person who is behind the discovery of this GUS gene is Dr. Richard Anthony Jefferson, and this gene is mostly used as a reporter gene in plants. So in this manner you will be able to see the transformed cells, the cells which are having this GUS gene. This is the rice embryo, and this is the anthers and style portion of this rice plant, and the alluron layer, the margins of this particular seed, they are also showing the expression of this GUS gene. So in this manner you will be able to screen the transformed cells by using this reporter gene called as GUS gene. Coming to the second example, reporter gene, green fluorescent protein, it's called as GFP. GFP gene, it is isolated from the jellyfish Aquaria victoria. A lot of work was done on this particular GFP gene, mainly by 3 scientists, Osamu Shimomura, Martin Chalfie, and Roger Seen. So for their work, these people were awarded Nobel Prize in Chemistry in the year 2008. So this is a bioluminescent protein, and it makes cells glow under UV light green color, which can be detected by fluorescence microscope. So it is commonly used to measure the level of gene expression by means of fluorescence spectroscopy, or by flow cytometry you can measure the level of expression of the transgene, or you can quantify the expression. So this particular protein, it glows in blue light at 395 nanometer and giving green fluorescence at 510 nanometer. So this is the way in which the particular green fluorescent protein, it is expressed. This is the mouse embryo, and these are the bacterial cells which are having this particular reporter gene. So in conclusion, this screening method, they are used for the easy detection of the transformed cells. So here you are using the marker genes for identifying the transformed cells. So there are 2 types. One is the selectable marker. Just it is used to know whether our foreign gene is inserted or not. You will be able to select the transformed cells from that of the non-transformed cells by using certain agents, selective agents like antibiotics or herbicides. The second type is the reporter gene. So along with the detection of the transformed cells, you will be able to quantify the level of the expression of the desired gene by using certain types of method like this flow cytometry, spectroscopic methods. You will be able to quantify the level of expression of the inserted foreign gene. With this, I conclude the session. Hope you enjoyed learning this session. Last but not the least, I profusely thank Commissionerate of Collegiate Education for providing this LMS platform to reach a large number of enthusiastic learners like you. Thank you one and all.